So I'm going to try to talk to you today about strategies for obtaining patents in the New World Order. I call this Avoiding the Darkness. It is on the patent eligibility of biomarkers and biologicals. So what I'm going to do, assuming I can figure out how to use this thing. Okay, yes, overview. What am I going to talk to you about today? I'm going to give you some general background on U.S. patents. We're going to talk about what the requirements for patentability are. That may be review for some of you, but I think it's important to sort of review it so that you know where we're going on this. We'll talk about some recent court decisions. These are three names you'll see again and again throughout this presentation: Prometheus, Myriad, and Alice. They are three recent Supreme Court decisions, and they are important if you're looking to get a patent or you're looking to start a company. The next step will be the U.S. Patent Office eligibility guidelines. So this talk is going to primarily concentrate on U.S. patents. And this is how the U.S. Patent Office has taken these recent court decisions and applied them. I'm then going to go over practical examples. So I'm actually going to give you some claims. We're going to talk them through. We're going to talk about whether you could or could not get a patent on this. So as a review, what is a U.S. patent? So a patent is a property right as defined by the claims. Claims is highlighted in a patent in the U.S. government, by the U.S. government. So I like to think of what the claims are as the words that define your invention that come at the end of the patent. So what these are, and the best image that I have is my children when they were small standing on the beach, and they would draw lines around the sand, and they would say, "This is my area and you can't come in." And that's effectively what a claim does with words. So it draws that line in the sand for what you view as yours and where other people can't come in. So the patent owner has a right, and it's for making, using, selling, offering to sell, or importing the claimed invention. Remember what's defined by those words. A patent does not give you a right to practice. So, as an example, my kids didn't really own everything on that beach. The state government might have had a controlling interest here, right? So it doesn't necessarily give you a right to practice. It's a limited time in the United States. It's 20 years from filing. It's that in most countries. So from the time you file a non-provisional patent application, you have 20 years. And in order to keep that right, you have to pay the government money. We have to pay the government money a lot, so we should be used to this. It is limited by geography. So as I talk about patent rights here, I'm talking about U.S. patent rights. And that's pretty much where we're going to concentrate. I can address patents abroad, Canada, Australia, Europe, if you want, perhaps after the talk. So what are the requirements for patentability? What do you have to have in order to get a patent? And something's patentable if it's novel. It's got to be new. It's got to be inventive. You have to have an invention there, important word. And then sufficiently described and useful. And I think those are particularly important to remember. So if you think of Thomas Edison making the light bulb, he would have the right to exclude others from making the light bulb, but it had to be a sufficient specification so that somebody else could take those words, the whole description in the patent application, in the specification, and then make that light bulb when the patent expired. So it really has to have sufficient written description. And what we're going to be talking about today is other tests, and that's a threshold test. Patent eligible. Is it something you can get a patent on? And I think here's the statute, 35 U.S.C. 101. Whoever invents or discovers a new and useful process, machine, manufacture, or composition of matter. So those are the things that are patent eligible, according to 35 U.S.C. 101. And that's what we're going to be talking about, patent eligibility. So from a historical perspective, patent eligible subject matter is anything under the sun that's made by man, pretty much. But, well, actually, let me take a step back. And are biologics patent eligible? Yeah, they are. And that goes back to a case, Diamond v. Chakrabarty, genetically modified microorganisms are patent eligible. So there are a lot of biological patents. There's a floodgate of them. And these are what have been patented in the past. Purified synthetic forms of DNA and proteins, methods in molecular biology, methods of diagnosis and treatment. You can certainly think of methods of surgery and dosage adjustment. Big field today, personalized medicine. And so this talk is going to sort of try to touch on all of these and the patent eligibility of these. And I'm really having trouble with this little clicker thing here, so you're just going to have to forgive me a little bit. And what has happened now is that there's been a huge controversy, and that's over the patentability of DNA inventions. And you've probably heard about this. It's brewed for years. It was never previously considered by the courts. And the controversy has brewed for years because there's a lot of people involved in this process. Anybody see themselves in there? Certainly judges, inventors, trolls, and activists, and all come into play here. So the troll guy, I don't know if anybody watched John Oliver last night. He did a really interesting thing on patent trolls. I thought it was kind of cool. It may have been a little simplistic, but I thought it was really cool. So I think he does a great job. So what happens? The results can be chaos. And what we're having now is really some upheaval in the field. So what's at issue now? Really, because of all those personalities that came into play, the law of what's patent eligible in the United States has really been challenged by the Supreme Court. And here are those words again, Prometheus, Myriad, and Alice, the three decisions. And then the Patent Office interpretation has come into play, and in my opinion, it's made a bad situation worse. So we have right now a little bit of an uncertain foundation. This is my favorite quote. Page of history is worth a volume of logic. So I'm going to go over the history for you, and then I'm going to try to give you the logic of the implementation of that history. So why is there a debate now? Basically, although you saw that statute, now not all processes, machines, manufacturers, or compositions are patent eligible. Here's my clever patent drafter. You'll see him appear a couple of times. So what has happened now is that judicially we have carved out certain exceptions. And those are products of nature, specifically genomic DNA, and that's the Myriad case. Laws of nature, which is biological correlations, that's Prometheus, and some abstract ideas, and that's Alice. You'll see a lot of patents that issued before 2012 with claims that you're going to say, well, those are really close. And the answer is yes, and some of these patents were used to preempt others from using a natural phenomenon or idea, but it's really changed. And so you can bring up a patent from before 2012 to 2014. You're going to see very different things from what you see now. How did all those claims get through the Patent Office? Well, it was different before, and things have really changed now. So let's start by going over those decisions of Supreme Court, Myriad, Prometheus, and Alice. The Supreme Court, as you know, is the highest judicial authority on patentability in the US. There's three cases. Prometheus, 2012. This dramatically reduced the ability to obtain meaningful claims on diagnostic methods. Myriad's a problem. It's pretty much upset 100 years of well-set precedent on natural products. And Alice is really a problem when it comes to software. These were all unanimous decisions. These changes are here to stay, so I think they're important, and I think it's important to know about them. The basic theme here is that patents to claims or methods that preempt the use, so they have to preempt a use, they're not going to be considered patent eligible. You're not going to get to over the threshold to where the USPTO, at least, is going to look at your patentability. So let's go over these decisions. The first one's Mayo. And here's the claim. I've distilled it down to sort of what I see as the essential elements of the claim. The Prometheus claim was a method of optimizing a dose for a drug. You administered the dose to the patient, and then you determined the serum level of a metabolite. And what they really had discovered was a cutoff. So they discovered the cutoff value. The cutoff was if it was less than 230 picomoles, but it doesn't matter what that cutoff was, then you increase the dose. And if it was more than 400 picomoles, you decrease the dose. The Supreme Court said that this claim is patent ineligible because it's merely an application. It merely, there's a law of nature there. If you give the drug, the marker exists. That's a law of nature. And so all you're doing is the drug is breaking down into known metabolites, and just knowing an optimized range does not bring you to patent eligibility. The claim steps add nothing of significance. It's important to note here that there's no treatment step. It's just medical decision making. So that's sort of a summary of that one. And we get to the beginnings of a test. Is the method focused on a natural law? If so, does it contain something more? than the natural law itself. And remember, in Prometheus, it was just a cutoff value. So to be patent eligible, the method has to add something that is more than what's well understood, routine, and conventional. We have to look at whether there's an inventive concept. Patent eligibility cannot depend on the draftsman's art. So it can't just be something mentally, something added. It can't just be a draftsman's art. So now we go over to Alice. And here's the Alice claim. It's kind of long, but again, I've distilled it down to sort of what was at issue. We had a computer-implemented method of managing settlement risk, and this included creating shadow records, obtaining start-of-the-day balances, adjusting the shadow records, issuing some sort of end-of-the-day instructions, and that led to another test. So as you can see, each of those steps, it's sort of a mental step, right? Creating, obtaining, adjusting, issuing. It's sort of something you could do in your head. And that's sort of a judicial exception. What else is there in the claim, right? It's just mental steps here. Do the additional elements transform the nature of the claim? And the answer was no here. So it was just a mental step. So the method claim was patent ineligible because it preempted the use of an abstract idea, managing settlement risk. It was something that could be done on a general purpose computer. But merely adding a general computer to an abstract method does not impart patent eligibility. And here's a good quote. In applying a judicial exception, we have to distinguish between patents that claim the building blocks of human ingenuity and those that integrate the building blocks into something more. And that something more is going to be a theme that you hear again and again today, something more. And once again, patent eligibility cannot depend on the draftsman art. So what is the impact of Prometheus and Alice for us? How does that apply here at Emory, or how does it apply to a company or at any other university where they're studying medical technologies? It applies, of course, for diagnostic methods. Drug discovery methods, think about it. You're screening something, you've got your cells in a dish, you want to know if your chemical compound is a good agent as an antagonist, you're looking at something that goes up and down. Wait a minute, you're just looking at the correlation between the compound and whether it goes up or down. Diagnostic software. So Prometheus and Alice really constrain patent rights involving any place where you're using a correlation, a biomarker detection. And the US patent standard, I want to mention this, is different now than most industrial legislations. These things are only at issue here in the United States. It is different everywhere else. And most people say that it makes us below everybody else. But it's really different. It's really changed. So what's the takeaway test from here? What do patents not cover? So to be a patent eligible process, you have to do more than simply discover the scientific principle and apply it. You have to do more than just discover the biological correlation. You can't claim the natural principle. You can't claim the mountain. No natural principles without something more. And this goes for a law of nature, natural phenomenon, and naturally occurring relations or correlations. So let's go over myriad genetics. Somebody today called it the breast cancer case. I think it's a good description. So patent eligibility of DNA inventions was challenged by the ACLU. It was a lawsuit against myriad genetics. The patents covered isolated genomic DNA, cDNA probes, primers, and the use of BRCA mutations to predict susceptibility to breast and ovarian cancer. And we asked a couple of questions here. Is isolated gDNA, genomic DNA, a natural informational molecule? Is it sufficiently or insufficiently changed from its natural state to make it patent eligible? And what about methods of diagnosis? Are those just mental steps? So again, I've sort of tried to distill the claims down to what's at issue. We had two claims here, isolated DNA encoding the BRCA protein, that's gDNA, and a synthetic DNA encoding the BRCA protein, a cDNA. So the Supreme Court considered this question. It's really a simple question. Are human genes patent eligible? And the answer is No, for some of it, isolated DNA having the same sequence as a naturally occurring DNA, genomic DNA, is not patent eligible. Doesn't matter if you break the bonds and take it out of the chromosome, it's not patent eligible. Yes, for synthetically created DNA, and here's a good quote from the case. Myriad did not create anything. To be sure, it found an important and useful gene, but separating from the gene is not a useful invention. So the mere act of breaking the bonds and isolating genomic DNA, even though you didn't know to isolate that piece of DNA, did not make it an invention. So what's the impact? So cDNA remains patent eligible. It's more commercially important probably than gDNA. And this holding was limited to DNA. But, and this is a big but, natural products, something you isolate from nature, not patent eligible. So I'm going to go over a couple of Federal Circuit cases that are out there because I think they're sort of illustrate what's going on a little more. The Federal Circuit is the court that handles the appeals of patent cases. It's a level below the Supreme Court. Here's claims held to be patent ineligible recently. A pair of primers for amplifying a BRCA1 gene. Well, those primers, they're 15-mers. They're useful for PCR, but they were identical in sequence to genomic DNA. Primers, not patent eligible. A method of detecting a mutant allele, and it just had comparing the sample with a control. No, that's not patent eligible. Methods of detecting a mutant allele of a BRCA gene by comparing a genetic sample from a subject with a control, and then using a probe that hybridizes or using amplification in DNA sequencing of the BRCA gene. Nope. All of those claims recited general methods. There were no specific method steps or reagents recited for these claims. You can see there's no specific antibody or anything in there. No specific alleles, probe sequences, primer sequences, but those claims are all patent ineligible at the Federal Circuit. Okay, so we have limitations on DNA patents. A discovery is not patent eligible. gDNA, informational content, but cDNA, okay, we've got something here, sufficiently dissimilar to gDNA created in the lab. So, am I going the right way? Minimal impact from specific holdings. Sorry, pointer issue. Genomic DNA is no longer commercially critical, but now we're going to see some greater impact from the implications of these cases. And this is the USPTO guidelines. So this is how the US Patent Office now is applying the holdings from these cases. I wanted to mention this. I threw in the slide because I think it's interesting, and I like clone things. So Dolly the clone sheep, this was a Federal Circuit case, not patent eligible in the United States. It was determined that Dolly is not sufficiently different enough from a natural sheep. Dolly was patent eligible in other countries, but apparently she's not in the United States. The claim at issue here just was to a cloned sheep. It did not mention the difference in the mitochondrial DNA, and so I want to throw that in because I think it's important that had that been mentioned, the finding might have been different, but Dolly the cloned sheep was not considered patent eligible. So the USPTO decided that they want to clarify patent eligible subject matter. They originally issued guidelines in March of 2014. There was a period open for public commentary. The public commentary was huge and predominantly negative. Revised guidelines were issued in December of 2014. They're actually a little bit more reasonable, although you may not think so after my talk, where structural and functional differences were considered. The guidelines as they stand now primarily address product claims. There has been a new public comment period, but it is closed. We're right now not recommending you make any rash decisions in this changing legal environment, but I think they're very important to know about. So what I'm telling you is that the public comment period is closed. We don't know if they're going to change the guidelines or not. They may change. We suspect it won't be hugely changed. So the Patent Office analysis, how do they look at your claim if you have something and you have a patent application and you put your claim in? How do they take a look at it? The first question that they ask is the patent claim directed to a process, machine, manufacturer, or composition of matter? And if the answer is yes, then they ask the question of, is it directed to a judicial exception? And that would be an abstract idea, a product of nature, or a law of nature. And then they ask if the claimed product is markedly different or clearly does not tie up the judicial exception, then they decide it's not directed to the judicial exception and it's patent eligible. And that means you get to be considered on all those other grounds. So if yes, patent eligibility requires the claim as a whole has something significantly more. So that's the word. So if the claim is directed to a judicial exception, they're going to be looking to see if the claim recites something significantly more. So I'm going to go over some examples so that you can sort of see how this is practically applied, because I think it's a lot of words and it's hard to see until you sort of start to see things from the lab brought into more of the reality. And what I'm going to try to do is give you ways to avoid the darkness, okay? So natural products. To be patent eligible, the product as claims has to have a marked difference. You keep seeing those words marked and significantly more from what exists in nature. But even minor differences from nature can make something markedly different. You can't base eligibility, remember the gDNA, on the mere recitation of purified or isolated. So we're going to consider compounds such as those extracted from natural products, naturally occurring microorganisms, and isolated proteins. Think of those. Because it's important. 50% of the FDA-approved drugs since 1980 are natural products or derivatives. Right? So these things are important. So let's go through our examples. Here's a first example. This is an example directly from the USPTO guidelines. So this is what they say. And this should look to you like the BRCA claim. An isolated nucleic acid molecule with a specified sequence. It has seq ID number 1. And claim 2 is an isolated nucleic acid molecule. It has sequence identity 90% of seq ID number 1. So seq ID 1's natural. Some of those other ones are not natural, right? Is that eligible? No. The claimed nucleic acid is not markedly different from genomic DNA. So it's not patent eligible. So claim 2 encompasses claim 1. It encompasses the naturally occurring sequence. That's not patent eligible, neither of those. So how could you design around that? Well, I suppose you could add a label. So if you've got a primer sequence, you've got a quencher and a fluorophore on it, something like that. Certainly a specific mutation, not found in nature. What about the DNA in the context of a particular expression vector? So those are always things that might impart patent eligibility to this. And I'm not saying a general expression vector, I'm saying a poxviral vector or an adenovirus vector, something unique. Problem is that makes it more easy to design around. So here's another example. We mentioned primers. A pair of primers that amplify DNA from an exosome. They comprise 2 sequences, seq ID number 1 and seq ID number 2. And there's no material difference between the primers and the naturally occurring DNA sequence, but they don't occur together, right? They don't occur together. Is this claim patent eligible? Nope. The 2 primers are identical to the naturally occurring DNA sequence. Here we go again. What are my solutions? Solutions are a label, a mutation, a primer that expands an intron, right? Because that sequence would not necessarily, you have intron exon border, so you've got part of exon 1 and part of exon 2 in your primer. A method of using the primers where you have specific steps in the method, a specific type of RT-PCR. Once again, your problem makes your claim more easy to design around. So this is a USPTO example, and this is literally their example. So an antibody to Staphylococcus texana protein. This staph is known to affect coyotes in Texas, which I guess is why it's called Texana. Infected coyotes produce antibodies to protein S. So the claims encompass a naturally occurring antibody. They could encompass non-naturally occurring antibodies, but they encompass the naturally occurring antibodies. So that claimed antibody is not markedly different from a natural antibody. So that one's not going to be patent eligible. Same solutions, add a label, a particular label, adding a linkage to a solid support. A particular mutation, you have an FC domain that increases your half-life of your antibody. You have a humanized form of that antibody. Those are probably going to be much more patent eligible. You want to ask a question? Go for it.
What if I designed this antibody by exposing rabbits to this toxin?
Well, that's a hypothetical example on the next slide. Oh, okay. So you're very smart. So it's okay. So that's an interesting question. And here we have an antibody that specifically binds biomarker X, and it has 6 CDRs, standard 6 CDRs with specified sequences. It doesn't matter what they are. And say the antibody is produced by immunization, which I think is what you're selecting, or it's suggesting, or it's selected from a phage display library. The USPTO views those claims antibodies as being markedly different from antibodies, from known antibodies, and it's produced in a lab. So that is patent eligible. So what the USPTO says, and I've talked to a lot of examiners about this, is that the production of an antibody is a stochastic event. You can't know what combinations of CDRs are going to be present in an antibody. Therefore, as soon as you force the situation, either by immunizing a rabbit or using Hugh Cow Gold, it doesn't matter how you got there, that one's going to be patent eligible. Humanized and chimeric forms also okay here. So the USPTO example, you notice it said antibodies to staph taxana, okay, and that was literally their example. But currently, we never get an antibody claim without specifying the CDRs. So I wanted to mention that because somebody might ask, like, well, the claims were really different. Well, really, we never see, almost never, claims in the United States that are an antibody that specifically binds protein number 1 anymore. They always have that structure. So that's not the difference here. And antibodies, it depends on where it's isolated from. So it's completely coming from how that antibody was produced, whether it was produced in your lab or in the claim. And I think I wanted to add the second note. Antibodies isolated from nature are not patent eligible. There's a huge patient population where you may have a proprietary patient population and you have isolated a particular antibody that you think is of use or important in an autoimmune disease, such as MS, because that's going to help you design your vaccine or whatever it is. If that antibody was isolated from that patient, that's not going to be patent eligible. It doesn't matter if you put the CDR sequences in it. Okay? You have a patient that's isolated, got a virus, and you want to transfer in the immunoglobulin. It's not going to be patent eligible. So if you want to isolate that immunoglobulin and then give it to another person, not patent eligible. So it's really limiting the antibody claims. Now, I think most antibodies that are of use commercially are produced the other way, but keep that in mind. The next example is a hypothetical example. It's an isolated stem cell. I have a lot of these cases too. It's an isolated stem cell. It expresses 3 markers, ABC. It proliferates for 20 population doublings. It occurs naturally in humans. Removal and purification does not change its structure. Okay, so that's your classic stem cell. We've isolated the stem cell. We want to give it to people. We want to use it. And click. That cell is not markedly different from a native cell. So that's not patent eligible. So if you're going to isolate a stem cell out of someone and thinking that you're going to give it to others, that's not patent eligible anymore. I will tell you that I have a case that I'm working on. It's not on this slide. It is a human ABC plus stem cell, and when you take it out in vitro, it expresses a new marker. 100% of the cells express a new marker. Not only that, the new marker seems to be what's allowing it to attach to plastic. I have a rejection from an examiner in 2 cases that are very similar. She is saying that that is also not patent eligible because the stem cell inherently will express that marker when you take it out and put it in plastic. So all of the stem cells will express that marker. So that's what is going to happen by definition. Therefore, you're preempting the entirety of that stem cell. I think we're going to appeal that one because it's not what's in the human body. But that's where I am on that. How about an isolated human ABC stem cell comprising vector X that proliferates for 40 population doublings? So I put in a vector. Now it's a transformed cell. It's not naturally occurring. It's got this interesting, oncogene or something in it. It has properties. That's going to be patent eligible. Now, whether that's what is of interest to you as a medical product, I don't know, but that is going to be patent eligible. So here we have another hypothetical example, an isolated exosome comprising biomarker X, isolated from a human without any alteration. You know that answer, right? It's patent ineligible. It's identical. So what's a solution here? A solution is a composition including the agent the exosome, and perhaps a stabilizing agent. So if you have a particular preservative and you're going to use that, and it doesn't matter if it's a natural preservative, that might be patent eligible. The additional agent somehow needs to give a functional difference to the combination. So as an example, if you have a vaccine and you're going to put a certain adjuvant in there, right, that it might get you to patent eligibility because those two things are not found together in nature, and they're going to give you a more robust immune response. And one of the USPTO examples now on stem cells has a stem cell on a gelatin sponge. And of course, a gelatin sponge is not natural, but even so, the stem cell differentiates into when it's on the gelatin sponge and say doesn't differentiate when it's on a collagen sponge. That's patent eligible. So think about combinations of things as you're looking to get a patent. Once again, the problem might be a little bit more easy to design around. You could use a different adjuvant, perhaps, with your immunogen. All right, yeah?
What if you use the biomarker to distinguish between diseases that normally wasn't used for or hadn't been used for before?
So the biomarker, so tell me, say it again.
The biomarker has been previously used for breast cancer, and now you say you can correlate for the disease now.
Still a correlation. You discovered the correlation, but it's still a correlation. I don't think the patent office is, I don't like necessarily what the Supreme Court and the USPTO decided, but I think even if it's a biomarker for a new disease, that's still just a correlation. You've discovered the existing natural principle of the correlation of that biomarker with that new disease. So, product claim, practice tips. Identify structural and functional differences between a natural product and what might be used in your therapeutic or diagnostic assay. So, an example, label proteins, label nucleic acids. Compositions that include several components. Your adjuvant, what are you going to put into the actual pharmaceutical composition? Identify genetic modifications that are of use. And an example here is cells transformed with particular vectors. But I think another example, I don't know if it's on this slide, it's structural changes that provide enhanced biological properties. So if you've cloned a new protein and you know the domains that are of interest, and you're going to mutate those domains and you think you can get enhanced activity, put them all into your patent applications. Put in those domains. Put in what you might change in detail. We file patent applications. We don't necessarily have proved everything. We have to believe it's going to work, and you have to be able to show that it's going to work later. This is true for US. Anything you can think of, put it in. Okay, let's go over some process claims. To be patent eligible, the process as claimed has to reflect a practical application of the law of nature. And here are those words again. Something significantly more than the biological correlation. So, as a general rule, if you could do all the active steps of the method in your head, it's not patentable. So, detection of a biomarker? No. If I could say, okay, it's up or down, no. Diagnostic methods with general steps, those are likely patent ineligible. Diagnostic methods with particular reagents or assays are likely eligible, but be careful. If I'm going to be claiming something with a particular reagent and you can do it with 10 different reagents, I can get a patent on it, but how valuable is that patent? Methods including a treatment step are likely going to be patent eligible. So methods of treatment haven't really been called into question here, okay? But we've got divided infringement issues. So if you say she's discovered a new, the marker is now with a new disease, and we say, okay, we're going to detect marker number 1, and this tells us if you have breast cancer or not. Doesn't matter whether it's breast cancer or uterine cancer, and therefore we're going to treat that person with this combination chemotherapeutic, all in one claim. That's probably patent eligible, but no one party is going to perform all those steps. You're probably going to send the sample over to the diagnostic lab who's going to do the diagnostic test, and then the nice physician over here in the corner is going to treat Mrs. Jones. And so there's not going to be one party performing all of those steps.
But in that case, is it not still considered combining the building blocks of previously established procedures? If you do it that way?
So we're talking about patent eligibility. Patentable, novel, new, and non-obvious, that's another question, right? So if those two things were already known, then yeah, you're right. But I'm assuming your marker is new, your test is new, right? So it's not something that's out there before. So there's no building block that's out there before. Okay, Here's a method of treatment, a USPTO example. It's a method of treating breast or colon cancer by administering an effective amount of purified amazonic acid to a patient suffering from breast or colon cancer. Amazonic acid is a natural product. This claim is patent eligible because it's focused on practically applying the natural product, and it's a particular disease. So it doesn't just say cancer, it says breast or colon cancer, and it would be even better if you put a dosage range in there. They'd be even happier with that. So what about with a method of diagnosis and that biological correlation, right? You could add that step. So here's a diagnostic claim. This is a hypothetical example. The USPTO has promised us that they're coming out with method examples. They promised us in January they'd be coming out in a month, and you can guess what's coming next is we've heard kind of the same thing, that they're coming, and they are not out yet. So most of these examples are hypothetical examples. So this is a method of identifying a subject with disease A. You isolate an exosome, you detect the level of a microRNA, and then you identify the subject as having disease A with that increase. So it's a diagnostic claim, general steps, biological correlation. Is that eligible? Probably not. What could you do? Add some limiting steps. Particular device that you use? Is there a particular device you have to use? Can you only detect this marker using a particular kind of mass spec, so you must use this device in the lab? I mean, it still exists, but you could detect it perhaps other ways, but it's not so great. Assays, if there are particular assays where you can actually have steps recited in your claim, reagents. Do you have to use a particular antibody? Do you have to use a particular heating and cooling step? These limiting steps all would have to be disclosed in your application, but moving them into the claim would help you. Once again, that's way more easy to design around, right? Okay, here's another diagnostic claim. A method of determining if a subject has lung cancer by obtaining a sample from the subject and contacting the sample with a monoclonal antibody. It's got the 6 CDRs, wherein the antibody determines that the subject has lung cancer. So that's probably going to be patent eligible, especially if that antibody was produced in your laboratory. If you have it recited as generally any antibody, probably not. So if it's any antibody that binds that marker, probably not. Okay, I'm going through this because it's a concrete example that we have. Here's the Myriad example. So they had a method for screening the genome of a human subject for alteration of the BRCA1 gene. They compared the BRCA1 genomic or RNA sequence from a tissue sample with a wild type. That didn't help them. Wherein the BRCA1 or genomic RNA sequence is compared by hybridizing, hybridizes to BRCA1 allele and genomic DNA and detecting the presence of the hybridization product. Nope. Wherein the BRCA1 genome is compared by amplifying using a set of primers. Note the primer sequence is not specified. And sequencing the amplicon, that didn't help them. General. So what could Myriad have done? Add limiting steps. That's really the only way you go. What kind of limiting steps? Particular probes, perhaps? Alleles, type of PCR assays, particular sequences? The first party with knowledge of the BRCA1 and BRCA2 sequences, Myriad was in an excellent position to claim applications of the knowledge, right? Many of its unchallenged claims are limited to such applications. So when you get to specific things, you're going to be much better off. So the unchallenged claims include references. So we don't know, but we think the unchallenged claims include a reference to specific BRCA1 allele, specific mutations, very specific probes for detection, and active steps. So in all of those steps that were before, they were a lot of sort of passive steps, so comparing by hybridizing. I think active steps help as well, and those are important to include.
Susan, what do you mean by unchallenged claim?
Nobody brought them up. Nobody brought suit against them against those claims. So they were not challenged by the ACLU.
So they stand.
They stand. So what are some practice tips when you get to process claims? Describe your methods in your patent application in excruciatingly detail. So maybe we won't start out with such a narrow claim, but we want to have things we can move into those claims. Really identify critical reagents. Really important to know what reagents we're going to use, right? And devices needed to practice your method. And then identifying non-natural products. Those would be good for use in the method. So if you have a method and it uses a specific type of PCR assay and it's got a quencher in it, say it. Put it in there if you need to. Of course, mutated proteins, non-natural agonists, anything that's non-natural that you have to use. And the bottom line is you really need to disclose multiple examples of reaction conditions and devices and identify the reagents that are critical to the process. I would put that into my patent application right now. I would start with the broadest thing that I could do, and I would make sure I have fallback positions in my specification, because we're not there yet. We don't know all the answers. Your claim scope is going to be more limited. So you can bring me patents from 2012 and 2013, and you could say, what about this, and what about this, 2014, what about this? And the answer is, it's changed. We're talking about guidelines that were issued December of 2014, and we're talking about a public comment period that closed in March of this year. So things have changed and are changing. So just because people got them in the past doesn't mean it's going to be that way now. The claim scope is going to be more limited, and this is particularly true for diagnostic methods. Treatment methods, I think, are still okay. So treatment methods still look okay. So I've been talking about US. What changed abroad? Really nothing. So all of these cases are limited to the US. To the extent that they were patentable, claims to all these things, diagnostic methods, methods of treatment, drug discovery methods, natural products, even DNA, those are still patent eligible everywhere else. So Europe is looking at us like we're nuts right now, in my opinion. But it's anything that you could get previously, it's untouched by these cases. So these are all US only. Okay, where do we go from here? So what are my recommendations for obtaining patents? Continue with your development activities, and I think the Brits have it the best here. Keep calm and carry on, right? So there's still a lot of things out there that are patent eligible. Keep on with those development activities and try to identify new ways that a natural product could be modified to give it a new activity. Try to identify things that you would change. Important to identify reagents and instrumentation that is critical to the process. It may be needed in those claims. Try to identify essential steps in the method. We want to make sure that our method is going to be more limited. We're going to have the temperature conditions in the method. We're going to have, it's a PCR method, we're going to have, it has to be this kind of Taq polymerase in the method. These claims are going to be more narrow now. I don't think there's any question that the claims that you can get are going to be more narrow now. So those are some basics, and it's not letting me go to the last one. And draft many claims, taking into consideration US and foreign guidelines. So I always like to have claims at the end that are an onion skin. If you think of an onion, it's got many layers all in a circle, going back to my circle image. We want to try to protect what you have with many layers. I don't think you're going to be able to get in the United States any diagnostic claim that's just measuring a particular marker. I don't. But we want to have many layers so that we could get perhaps with a certain antibody, with a certain device, how to get it around it as many ways as possible, going from the broad to the narrow. I think you also, when you draft a patent application now, you have to think, is this just for the US or is it just for foreign? Because in abroad, a claim to a method of detecting disease X using biomarker Y, that's going to be fine as long as you've discovered a new correlation. So I think we also have to start thinking beyond the US. There are different sets of rules for every country. So the standards are different in each country. They have different ways that they look at them. It's becoming closer to more unified. Right now, US is an outlier with this. So when you get to a diagnostic method, that is patent eligible in Canada, pretty much any country, well, not pretty much any country you can look at, the big ones that you think of, Canada, Australia, and European countries, they're patent eligible there. India has its own issues. China has its own issues. So the laws and rules and regulations are different in each country. The way that patents usually go is you file in whatever country you're in. There's something called a PCT, an international application, which is a placeholder application in all countries signatory to the Budapest Treaty. And so that's a placeholder in all those countries, but ultimately you have to go into each country individually. The costs are high when you're doing that. They're quite high. And yeah, they're really specific to the country's borders. So when you write your patent application, that all has to be disclosed in the text. So you can't add new matter later. Now, if it's there and it's not in the claims, you can move it into the claims. So what I usually get as an invention disclosure is a broad general description in a paper. And whoever is the counsel should then, once that decision is made to proceed with that patent application, would then glean all that information from you, because otherwise the invention disclosures become very cumbersome, and sometimes the university doesn't have the funding to proceed on every single invention disclosure that they get. So I would say put in, I can identify reagents or devices and would be pleased to do so if you want them. That's what I would do. You never know what's going to happen in a court in a certain country. So in Europe, they have very specific laws and cases that have come through on the patenting stem cells. And what they say is that you can patent a pluripotent stem cell, but not a totipotent stem cell. So they're very against the ES cell technology. And that doesn't come here. But that in Europe, those kinds of questions are far more important to them than whether a diagnostic test is patent eligible. I really think, and I went to the EPO, when was that? In January. And I actually talked to one of the people on the board, and I was asking them what they thought. So these were people who were on the board at the EPO on this board, and I said, what do you think of our cases? And they just sort of rolled their eyes at me. Patent examiners, yes. I mean, certainly patent examiners aren't premier researchers at Emory University, but they're sort of limited by what the rules are, and they sort of just try to follow the rules. And so their rule says, if it says detecting marker number 1, they don't know that there in the future isn't going to be a new way to detect marker number 1. They can't possibly predict that. So if you're going to say detect marker number 1, and you're going to argue to them there's only one way to do this right now, they're going to say, but there might be another way in the future, and you're going to preempt all future ways of possibly detecting marker number 1. So if there's only one way to do it, then put those words in your claim. Nobody else can infringe your claim. There's only one way to do it. Put the words in it. But the words have to be present in the specification in order for you to move them into the claim. So I would tell you to disclose it. The other piece here is that we're talking about patent eligibility. The rule really is there has to be sufficient written description and enablement for somebody of skill in the art to make your invention. You're required to do that. So you're required to give your postdoc, think of it as if you had a postdoc in the lab and you now want them to go and do that test. There has to be sufficient written description for that postdoc to run out and do the test. And if you don't tell them how to do the test, then your patent application might be deficient on that grounds too. The funny thing is you get assigned a patent examiner, and it's like a judge, okay? And you know that you can go into courts, in different courts, and the judge has there are different judges. There's one who hates everybody who ever commits a crime, and he's going to throw the book at you. And there's another one who's going to look at you and go, oh, you're a 20-year-old kid, and you got a little drunk, and it's okay. I'll make you do 100 hours of community service. So patent examiners are different, okay? And what one patent examiner sees, and I don't like to say this because people want, oh, this is the answer. Usually if you put enough steps in, you'll get by most patent examiners. So usually if you've got limitations, if it just says heating and cooling, you're probably not going to. I can give you probabilities. If it says heating and cooling to 45 degrees and cooling to 20 degrees, that probably is. A range might get you there. I mean, it really depends on the examiner what the technology is. So in some things, you're right, what's just well-known and conventional isn't going to get you there. So saying performing PCR by amplifying the gene, that will not get you there.
But I could go into the PCR.
And so one of the patent office examples that they first came out with was they had PCR conditions and they said using Taq polymerase. And they said, well, that's okay because you could use other polymerases. Now, in practice, I don't think that would work. So when I actually get to most examiners, that doesn't work. There's also the question of whether the claim that comes out is enforceable. So as much as you can get something through the patent office, you also want something that couldn't be challenged. And I think if it were just general steps from a PCR manual and it did get through the patent office, I think somebody could challenge it. And I think that's another issue for you.